Hi, I'm Olivia, and for the next 5 minutes, I'll be introducing you to your endocannabinoid system. Live from the Flaming Mountains in the Xinjiang region of China, a middle-aged man has been found with 800 grams of cultivated cannabis with high THC content. Hold on a minute. This isn't a scandalous news story about a tourist facing a prison sentence far from home. It's actually the findings of an archaeological excavation of a grave dating back to 750 BC. You see, humans and cannabis have existed in mutually beneficial symbiosis for the last 12,000 years. After the last glacial age, cannabis seeds followed human nomadic migration. Cannabis provided fibers for ropes, nets, food, and seeds for oil. Likely by chance, our ancestors will have discovered the euphoriant properties of heated cannabis and the resin produced by the distinctive female plants. Humans then began to cultivate cannabis, selecting for either psychoactive or fibrous properties. But it wasn't until the 20th century that the science of cannabis started to become clear, with the isolation of at least 113 cannabinoids. For example, you might have heard of CBD and THC. It was the determination of the structure of THC, the primary psychoactive cannabinoid, by Mechoulam and Guyoni in 1964 that incited the investigation of an unexplored neuromodulatory system, the endocannabinoid system. As it turns out, your body is filled with endogenous cannabinoids, or endocannabinoids, even if you've never even heard of cannabis. And this system is crucial in maintaining homeostasis, the maintenance of a stable internal environment. In fact, studies have suggested that there is endocannabinoid influence in appetite, digestion, immune function, inflammation, mood, sleep, reproduction, motor control, temperature regulation, memory, pain, and reward. And this list keeps growing as research progresses. The endocannabinoid system comprises the endocannabinoids, their receptors, and anabolic and catabolic enzymes. The two principal cannabinoid receptors are CB1 and CB2. CB1 receptors are expressed primarily in the central nervous system, whereas CB2 receptors are thought to be expressed within the peripheral nervous system and immune system. This being said, studies of CB1 and CB2 knockout mice have found cannabinoid responses independent of these receptors, indicating that cannabinoid reception is still not completely understood. The cannabinoid receptors are activated by the endocannabinoids, the most studied of which are 2-AG and AEA. Despite sharing the same receptors and similarities in chemical structure, AEA and 2-AG are different as a result of the different enzymes controlling their synthesis and degradation. But how does this all work? The endocannabinoid system is so effective in the maintenance of homeostasis as a result of its capacity for retroactive signaling and rapid liberation. To understand these specialized capabilities, we have to first understand neuronal signaling. Within the human brain, there are approximately 86 billion neurons, and most of the communication between these neurons occurs at structures called synapses, a gap between neurons small enough that it facilitates chemical communication. The presynaptic neuron houses chemical signals called neurotransmitters, such as dopamine and serotonin. These neurotransmitters are packed into vesicles, and when triggered by an electrical signal called an action potential, the vesicles fuse with the presynaptic membrane, releasing the neurotransmitters into the synapse. These neurotransmitters then bind to receptors on the postsynaptic membrane, triggering a change in the postsynaptic neuron. This can be excitation, an increase in the likelihood of generation of an action potential, inhibition, the opposite, a decrease, or initiation of a second messenger cascade. The neurotransmitters then drift away by a process called diffusion, are reuptaken by the presynaptic neuron, or may be degraded by enzymes. Retroactive signaling is signaling directed backwards from the postsynaptic neuron to the presynaptic neuron. This is where rapid liberation becomes important. If the neurotransmitters released from the presynaptic neuron taken up by receptors on the postsynaptic neuron are inconsistent with homeostasis, endocannabinoids can be rapidly generated and released to bind to receptors on the presynaptic neuron to trigger a change in neurotransmitter release. Through a range of different downstream pathways, these changes in neurotransmitter release maintain homeostasis, keeping our bodies stable. So there you have it, the endocannabinoid system, a widespread and highly influential neuromodulatory system discovered just 20 years ago. Many questions remain, but if recent research is any indication, one thing is for certain: this won't be the last you hear about the endocannabinoid system.